So welcome back, and we are in chapter 15 here. So let's change that. Chapter 15. And we're now reviewing the anatomy of the autonomic motor pathway. And in this figure from your textbook, you are looking at, look, they finally got it right. They have the cell body inside a lateral gray horn. And you know this is the parasympathetic nervous system because the color's red here. But more importantly, down here inside the key, it tells you it's the parasympathetic nervous system. Okay, so 30 seconds into this, and I'm realizing I need to remind you that we are in the second part of chapter 15. This is Bio 211 at Norwalk Community College. And again, these two sets of sort of adaptations of material from your textbook are no replacement for reading the book. This is me trying to help you understand the text so that you can apply it. And this is a great way to start right here, because look, if you follow the rules, you know all preganglionic neurons are myelinated neurons, and they're going to exit through the anterior ramus of the spinal cord. And in this case, it's going to use the pelvic splanchnic nerve, which fulfills the rule for our cranial sacral portion, because here's the sacral portion, and we're in the sacral portion of the spinal cord here. And you're going to see that the postganglionic C-type fiber is going to actually have its location in its target tissue. And for the purposes of this descriptor, we're going to say it's the smooth muscle of the urinary bladder. And what we can do as well, pick up the pen, now go back, use this as review for your exam at the exact same time. Change the color because we want to be consistent. We're going to go from blue to red because we're using the parasympathetic nervous system here. So you can tell me now that that is an A-type fiber, and that's the preganglionic neuron, right? And you know that then this is a C-type fiber. Put the arrow towards it. And that is the postganglionic neuron. And now we can start applying. How does Mother Nature differentiate these? Well, previously I showed you that all preganglionic neurons utilize acetylcholine to activate their targets. So we know acetylcholine is going to be used here, ACH here. And because this is the parasympathetic nervous system, we know the synapsing of that postganglionic neuron is going to use acetylcholine here as well. And you'll notice that there's no sympathetic innervation here. That's completely fine for our conversation. But this is a good review, and this is one way to start thinking about how it is you actually apply all this information. So how does Mother Nature actually ensure that you actually get all of the types of cells activated properly? We're going to change the numbers and the types of neurotransmitters and receptors that are available for activation inside the autonomic nervous system. And what they've done for you here is in part A, they've given you, said, here are the general rules for the sympathetic division for most effector tissues. Don't know what I did there, but let's do that. Most effector tissues. They give you one example, like I did in class, of how you would activate, say, a sweat gland using the sympathetic nervous system. Okay, so acetylcholine will activate this neuron here, which will release acetylcholine and will activate muscarinic receptors. For sweat glands, okay, for sweat glands, this is an exception. It is not the rule. The rule for the parasympathetic division is that you'll have acetylcholine binding to a specific receptor. That postganglionic cell will now release the neurotransmitter, that acetylcholine, and it will bind here a receptor. Okay, and you're going to notice inside this figure they've started naming them for you. So let's go back and take a look. Pick up the pen. I'm going to change colors to black here so that we don't get anything confused. Hey girl, my cat's about to visit us. Sorry about that. Okay, and oops, didn't change. Cool. So first thing I want you to see is that that cell body has nicotinic receptors on it. This cell body has nicotinic receptors on it, and this cell body has nicotinic receptors. And what we're doing is we're just simply reminding you that acetylcholine is made, and it binds to nicotinic receptors on the postganglionic neuron. Okay? So the general rule applies still. Everything we taught you is hunky-dory. The difference here is In the sympathetic nervous system, they're layering on this piece of information that says if norepinephrine has been exocytosed, it's binding a specific type of receptor. And these are adrenergic receptors. Okay, and I went over them in class. I'm just going to put an arrow down here for you. You have alpha 1, alpha 2, not alpha 3, beta 1, beta 2, and beta 3. Generally speaking, alpha 1 and alpha 2, beta 1 and alpha 1 are excitatory. Alpha 2 and beta 2 are generally speaking inhibitory. But generally, this means there's going to be exceptions, and we don't expect you to know all the exceptions. We just expect you to understand there's a pattern here. We use beta 3 for thermogenesis. This is increasing body heat, BH, in youth, meaning brown adipose tissue when you're born. Down here, you can see that we have a nicotinic receptor. Whoops, that's not what I wanted to do with that. Let's do that. Let's see if it erases everything. And it did. Sorry about that. But down here, you can see we have nicotinic receptors here. You can see we have nicotinic receptors here. And you can see we have muscarinic receptors here. And what this means is for us to excite our postganglionic neuron, we're using the nicotinic receptor. If we wanted to examine the types of receptors that acetylcholine can bind, you need to understand that there is muscarinic, MUSC, and nicotinic, NICT. And generally speaking, muscarinic is inhibitory and nicotinic is excitatory. So now if you put all those pluses and minuses together and you decorate them across all these targets, you can see how Mother Nature can have a multitude of effects on tissues. Okay, so let's re-put this stuff up here. You can see that we have, let's go alpha 1, and I'm going to put them together here, beta 1. Generally speaking, these are excitatory. How about alpha 2, beta 2? These are generally speaking inhibitory. And what you can do is you can begin to sort of understand that these are the receptors that you find on the cells, and if Mother Nature just simply makes the neurotransmitter available to bind them, there'll be an effect on that cell. Which is great for us, because what we want to do now is we want to think, if that's going to happen, how and why? And I'm going to walk us through a couple of tables that you find inside your textbook. Okay? The first is going to be nicotinic and muscarinic receptors. Okay? And what I want you to see here is that you actually have a type of receptor. This is how we name them. They are cholinergic receptors if they're binding acetylcholine. And the nicotinic are going to be excitation type, essentially. And you're going to see that generally speaking, the rule is nicotinic receptors cause excitation. You can, if we're generally speaking, having muscarinic receptors being used. In some receptors, you're going to see that it's excitation, and in others, it is inhibition. Okay? So what Mother Nature is doing here is now take a look at the examples. You can see that, say, let's use one here, that sweat gland. Okay? Sweat glands that are innervated by cholinergic sympathetic postganglionic neurons, so that middle structure I showed you a couple moments ago, okay, will increase sweating if the muscarinic receptor is activated by acetylcholine. So how do we apply this? Let's first compare it to the ANS, okay, meaning the adrenergic receptors, alpha 1, alpha 2, beta 1, beta 2. And, you know, I want you to read this. I definitely do, but I don't want to sit here and go through all the examples for you. I guess I'm looking at something that might be important. Cardiac muscle fibers might be important. Hepatocytes might be important. But don't try to memorize this. Try to understand what's actually going on here. So if alpha 1 has been activated, you have excitation. And the excitation here causes vasoconstriction. Okay? Let's look down here. Cardiac muscle fibers. These have beta 1 receptors on them. Excitation increases the force and the rate of contraction of the heart, obviously, right? Let's take a look at those hepatocytes. If beta 2 has been activated inside hepatocytes, remember that's epinephrine and norepinephrine, glycogenolysis occurs, which means you're going to break down glycogen into glucose. Think back to the first exam, right? When we were talking about how it is we make glucose available inside the bloodstream. And we make glycogen Okay, remember we store that as the polymer of glucose. Well, I'm teaching you now that if epinephrine, norepinephrine binds the hepatocyte receptor, beta 2, we're going to increase the amount of glucose inside the bloodstream. Okay, so that's how you want to look at these tables. They're more for interest than they are anything else. That doesn't mean don't read them. You want to have a sort of like, I understand how this works moment. Generally speaking, sympathetic and parasympathetic systems are antagonistic to one another. And I know that's obvious. At least I have to hope that it's obvious to us, right? What I want to do is finish this set of slides off with probably a couple of examples of how Mother Nature puts all of this together, right? So, sympathetic, parasympathetic, nudge, nudge, hint, hint, wink, wink. This is a really good table inside your textbook that helps you understand the basic pieces of information. I would go so far as to suggest you just simply write this whole thing out if you have the time, because you need to be able to access it during the exam so that you can, meaning it's inside your memory, so you can think your way through problems. But now, go down here. I want to, if this isn't found inside your textbook, what I want to make sure you understand, let's go back to the pen and go back to one of the drawings I put inside your class. I put inside class for you guys, right? So remember, all we drew when we introduced the idea to you is you actually had heart tissue, you had glands, and you had smooth muscle tissue, right? Not exactly Rembrandt here, but that's okay. So, and remember, we're going to innervate these with either sympathetic in blue. Remember, sympathetic, twang, twang. And if we're innervating them, chances are this is the postganglionic neuron, right? Remember, it's unmyelinated. And remember, we're going to have our postganglionic parasympathetic nervous system as well. They're arriving from slightly different locations, but for the expediency of the drawing experience, this reinforces the fact that it's antagonistic. And now what you want to do is you want to remember that, let's go back to black here. Okay, on here you could have muscarinic or nicotinic for all of the postganglionic parasympathetic targets. Or you could have alpha 1, beta 1, alpha 2, beta 2. Oops, let's do this. Wait till that stops. Beta 2. And now inside your mind's eye, what you want to do is you actually want to decorate each of these possible cell targets, because now inside the back of your mind, it's not just that you have a heart. Well, you got blood vessels on that heart. You've got other targets as well. You have adipose tissue here. So, it is important to understand the sort of basic underlying idea, but there's some complexity involved in as well. Thinking about this while I was just drawing this, I want to make sure you guys remember, if you go back to the cartoon of the sympathetic versus parasympathetic, remember the preganglionic neuron of the parasympathetic nervous system is much larger than the postganglionic neuron. Having told you that, I also want you to remember that most of the ganglia for the postganglionic neurons are actually at the target tissue itself. Okay. So what do we actually have here? This is a continuation of that previous table, and I could draw the whole thing out again, but here's where I want you to see how Mother Nature actually makes things complex. And I'm not showing you this so you panic and think to yourself, I have to memorize this. But what I want you to see is decreased motility and tone, okay, would be, if we've activated sympathetic stimulation, you would use alpha 1, alpha 2, beta 2 receptors. Okay, so it doesn't follow the general rule of alpha 1 and alpha 2 being excitatory. Alpha 1 and beta 1 being excitatory. Here you can see decreased motility of tone, which means when you're running for your life, sympathetic stimulation, you shut down motility and tone inside the small intestines and stomach. Okay? But if you activate it, you get increased motility and tone. This is where they get the rest and digest crap from. Okay? You're digesting when the parasympathetic nervous system has been activated. You stop that when you're running for your life. Okay? Although I don't want you to spend too much time thinking about this, the sex organs of the body, you remember at the cortex of the brain, you remember that the motor and sensory portions are disproportionately identified, meaning there's no somatic motor output for the reproductive structures. That's because the sympathetic and parasympathetic nervous system actually regulate for both males and females. Parasympathetic is the activation of the possibility of sexual activity, and the sympathetic nervous system is what allows for the actual, in this case, the males that they're talking about here, is going to be the actual allowing of the actual spermatocytes to actually exit the body. And notice what actually regulates ejaculation is A1. Now, this is one example inside your text. I don't know why they don't discuss the female responsibilities here. It's not that there aren't any. Okay. I suppose you could say that vasodilation starts for both males and females, and that causes the potential likelihood. But smooth muscle contraction, which isn't discussed here, has absolutely nothing to do with making spermatocytes available. But when spermatocytes actually touch the vaginal lining, you actually cause smooth muscle contraction. There'll slowly be some endocrine responsibility that we'll talk about in AMP2. Okay. Let's get rid of that. Let's get to the last table. In fact, I'm going to just go over here. And here it is. So you can see that, again, we actually have this general idea that if we actually have some sympathetic response, whoa, what did you do there, Jonathan? Let's go back over here. Sorry, my reflex time on this is not so good. Okay, cool. And what I wanted to do is just take a look at the heart and the arterioles inside of the heart. If you ever wondered why I said, look, we give you a basic introduction of how it actually happens. Well, relaxation of the arterioles inside the heart requires beta 2, whereas contraction requires alpha 1 and alpha 2. Contraction for vasoconstriction will utilize muscarinic acetylcholine receptors. Nobody's ever explained that to you before, but generally speaking, parasympathetic stimulation will have vasoconstriction. Okay? So how does all of this happen? Well, Mother Nature decorates the cells differently. So remember, when you're thinking about your cartoon, okay, and we're drawing lines today, just for time's sake, we're over 17 minutes, so I just want to finish this up, right? We just want to go like that. Remember, there are two that are working antagonistically. Because one of these is releasing, say, acetylcholine, that would be a cholinergic neuron, okay? And if this one is releasing norepinephrine or epinephrine, this is going to be an adrenergic neuron. But that would mean that the receptors for each of these are found on both of them, okay? It's just dependent upon where. And that's how you can differentially regulate the entire organ, okay? So that should finish it up for chapter 15. I'm going to go now and do some review materials for your next exam. Thank you for your attention. Have a great night.